Let's take a look at your graphic organizer for Module 9. You can locate your graphic organizer in your Biology Student Handbook at the end of Module 9. In Module 9, we first learned about how we can study human genetics using a technique called a pedigree. Over on the left side of your graphic organizer, you have a place to define what is a pedigree. So go ahead and write in the definition. A pedigree is used to study the inheritance of a single trait in a family. Now we're going to look at several different examples of pedigrees. And in order to understand these examples, you definitely need to understand the symbols that are used in pedigrees. Squares represent males, circles represent females. We are looking at a single trait, so individuals who have that trait are going to be shaded in with a darkened square or circle. If they don't have that trait, it will be a not darkened square or circle. A square and a circle that are connected by a line represent two individuals who are mating. When there is a line extending from that connection, that represents their children. You'll also notice on the left side of each pedigree, there are Roman numerals. Those Roman numerals represent the generations. Generation 1 would be, for example, your parents. Generation 2 would be you and your brothers and sisters. Generation 3 would be your and your brothers and sisters' children. Pay careful attention as we go over the different examples and make sure you clearly understand how these pedigrees represent the inheritance of those traits. Now, looking at the first pedigree, you will see that there are affected individuals in every generation, darkened circles in every generation, generation 1, 2, and 3. This is an indication of a dominant trait. So please write under example 1, autosomal dominant trait. Autosomal simply means that this trait is inherited on one of the first 22 pairs of chromosomes rather than on the sex chromosomes. In this case, and you need to write these bullet-pointed notes in the box provided, so write small, individuals need only one dominant allele in order to express the trait. In other words, if we are using the letter B, they could be big B, or big B, little b, and still have the trait because it's a dominant trait. We recognize this as being a pedigree for a dominant trait because the trait is present in every generation. We recognize this being an autosomal trait rather than a sex-linked trait because it is expressed equally in both males and females. For example number 2, looking at the pedigree, you will see that there are no affected individuals in the first generation. This is an indication that this might be a recessive trait. It has skipped a generation. It has skipped the first generation. So go ahead and write in on the line for example 2, autosomal recessive trait. In the box provided, write the bullet-pointed information. For a recessive trait, individuals need 2 recessive alleles in order to express the trait. For example, if we're using the letter T, they would need little t in order to express the trait. We recognize a pedigree for a recessive trait because it skips a generation. We recognize this being an autosomal trait because, again, it is expressed in both males and females. Looking back at the first generation, we say that those two parent individuals who are unaffected are carriers for the trait because they have produced a son who is affected by the trait. The last type of pedigree that we are going to look at is for a sex-linked recessive trait. So go ahead and write that on the line for example 3, sex-linked recessive trait. Looking at the pedigree, you can see first that the trait skips a generation. So that is your indication that this is a recessive trait. However, you'll also notice by looking at the pedigree that all of the affected individuals are male, and that gives you an indication that this is a sex-linked trait. In the box, you need to write the bullet-pointed information. For a sex-linked trait, males only need one recessive allele, because sex-linked is X-linked and males only have one X chromosome. So for example, if we are using the letter H to represent the trait, males who have the trait would be X little h y. Females, on the other hand, if they get the trait, would need two recessive alleles because they have two X chromosomes. For example, an affected female would be X little h, X little h. Because females have two X chromosomes, they can carry a sex-linked trait. For example, if they have the genotype X big H, X little h, they are normal and unaffected, but could pass the allele for the trait to the next generation. Once again, we know this is a recessive trait because the trait skips a generation. There are no affected individuals in the first generation. And we know this is a sex-linked trait because it is expressed more often in males and is inherited from mother to son because the mother passes her X chromosome to her son. The son receives the Y chromosome from his father. Now we're going to be looking at some traits which can be normal characteristics, which you've learned about previously, but certain traits are actually genetic disorders. Some genetic disorders can be diagnosed using a karyotype. You have a place to define the word karyotype. So write in the definition a picture of the chromosomes. You have a symbol of a karyotype on your graphic organizer, and there's one shown here on the slide. Notice that the first 22 pairs, there is a bracketed area to label this, are called the autosomes. Please label that in the bracketed area. Autosomes are the first 22 pairs of the human chromosomes. Then you will notice that the last pair of chromosomes is boxed in, and there is an area to label this as the sex chromosomes. The 23rd pair are the sex chromosomes. This pair is what determines whether an individual is male or female. This individual, because there are 2 X chromosomes, would be female. Now look over on the right side of your graphic organizer. You're going to fill in the blank. A karyotype may show chromosomal disorders, which are also called aneuploidies. Underneath the word aneuploidy, in parentheses, write an abnormal number of chromosomes. If there are fewer or more than the normal 46 chromosomes, speaking of humans, that is considered a chromosomal disorder, also known as an aneuploidy. There is nowhere to record this information on your graphic organizer, at least specifically, but it is very important that you recall how chromosomes for a karyotype are obtained. Remember that every cell contains a complete copy of the chromosomes. So we can obtain a copy of the chromosomes by simply extracting the DNA from one single cell. This is easy to do for an adult or even an infant, but sometimes we want this information for a developing fetus. We want to know, for example, if the fetus is going to be born with a chromosomal disorder. This also gives us very accurate information about the sex of the fetus, whereas a sonogram can be misinterpreted. A karyotype, because you are specifically looking at the sex chromosomes, is a definite answer if the fetus is male or female. There are 2 methods to obtaining fetal cells. The first one is called an amniocentesis. You can see the picture. This is when the amniotic fluid around the fetus is withdrawn. The amniotic fluid would contain cells of the fetus from which the DNA could be extracted. The other method is called chorionic villi sampling, or CVS. In this case, a vaginal catheter is used to go in and snip off a piece of the placenta in the mother. The placenta is composed of the same chromosomal makeup as the fetus, so those cells can be analyzed and their DNA examined. Now let's start looking at some specific genetic disorders. The first type of disorder that we're going to look at are the gene disorders. Now these gene disorders, as is already indicated on your graphic organizer, cannot be seen on a karyotype. Go ahead and write in just beside that statement that these disorders involve the inheritance of a faulty gene. You will recall that a gene is a section of DNA that codes for a particular protein. And of course, proteins determine your traits. So a single gene cannot be seen by looking at a karyotype. A karyotype only shows us the entire chromosome. So we're going to begin to fill in the chart on gene disorders. The first gene disorder is called Huntington's disease. Under inheritance, please write autosomal dominant. This is a gene that is not sex-linked and is a dominant allele. So individuals who have the genotype big H have the trait. Individuals that are big H, little h have the trait, but individuals who are recessive, little h, do not have the trait. Under the area for description, please write in what is shown here. It appears after the age of 40. So you can be born with Huntington's disease and never know that you have it until you begin to show symptoms after the age of 40. This disease leads to gradual deterioration of the brain. You can see the normal brain on the left and the Huntington's disease brain on the right. Eventually, this will lead to death of the individual. And as with any other gene disorder, there is no cure. The next gene disorder on your chart is sickle cell anemia. Under inheritance, write autosomal codominant. This trait is inherited through two codominant alleles. It has to do with your red blood cells. Individuals who have the genotype SS have the disease. Individuals who have the genotype SN have what we call the trait. Some of their blood cells are affected, but others are not. Individuals who have the genotype NN do not have this trait. They have normal blood cells. You can see on the picture what we mean by normal blood cells and sickle-shaped red blood cells. Write in the description, sickle-shaped red blood cells lead to poor circulation and pain. This disorder is predominant in African and African American populations. This is not necessarily a fatal disorder. The pro football player Tiki Barber suffers from this disorder. The next gene disorder is cystic fibrosis. Under inheritance, write autosomal recessive. So this means an individual who is homozygous dominant, big C, is normal. An individual who is heterozygous, big C, is normal, but they carry the trait. An individual who has the disorder has two recessive alleles, little c. In the description, write, this leads to mucus buildup in the lungs and the digestive tract. It is usually fatal. Some of the children who are born with this disorder live into their 20s, but not many survive past that point. This disorder is predominant in Caucasian populations. As with any genetic disorder, there is no cure. There are treatments. You can see on the left how the lungs are producing an abnormal amount of mucus, making it difficult to breathe. This also interferes with the digestion of food. You can see on the right a boy who is receiving a treatment. It's a vest that shakes his lungs and breaks up that mucus so that he can breathe more freely. The next disorder is called Tay-Sachs. In the spot for inheritance, write again autosomal recessive. So similar to cystic fibrosis, big T is normal, big T is normal, but carries the trait. And in order to have the disease, there must be two recessive alleles, little t. This is a very sad disorder. Under description, write, this breaks down the central nervous system, leading to death by age 4. Again, there's no cure, and death of these infants is imminent. It is predominant in Jewish populations. The next genetic disorder is called phenylketonuria, but is abbreviated PKU. It also is inherited as an autosomal recessive. So record that in the chart. So once again, big P is normal, big P is normal but carries the trait, little p has the disease. This is an interesting disorder because it can be controlled by diet. With this disorder, individuals are not able to break down the amino acid phenylalanine. So it builds up in the brain and leads to severely decreased mental function. However, if you do not eat foods that are high in phenylalanine, this disorder can be controlled. You can see on the chart some of the foods that are very high in phenylalanine and some of the foods that are low in phenylalanine. Because this disorder is easily controlled by diet, all babies that are born in the United States have a heel prick test to determine if they have this disorder. We don't want to begin giving them a milk-based formula if they do have the disorder, because milk is a food that is high in phenylalanine. Make sure you have recorded the description in the space provided. The next gene disorder is hemophilia. Please record the inheritance as sex-linked recessive. So this means individuals with dominant alleles are normal. Females would have X big H, X big H. Males would have X big H, Y. Females can be carriers of this trait because they have two X chromosomes. A female carrier would have the genotype X big H, X little h. In order to have the disorder, the individual must have recessive alleles, X little h, or X little h, Y. In this disorder, there is a low production of blood clotting factors. This means individuals with the disorder can bruise very easily and have excessive bleeding from wounds, including internal bleeding from bruises. You can see a joint bleed in the picture. It looks painful. Make sure again that you have recorded the description. This is not necessarily a fatal disorder, but one that must be managed. The last gene disorder that we will look at is red-green color blindness. This is inherited as a sex-linked recessive as well, so record that in your chart. Once again, dominant alleles are normal, X big B, or X big BY. Having a heterozygous genotype in a female is a carrier, X big B, X little b. In order to be colorblind, the individual must have recessive alleles, X little b, or X little by. Write the description: an inability to distinguish between colors, especially reds and greens. On the left is a picture that shows a test for red-green color blindness. If you are not colorblind, you should be able to see the number 2 in that circle. If you are colorblind, you probably can't see the number 2. On the right is a picture showing what the jelly beans might look like to a colorblind person. They see color, but they can't distinguish between various colors. Now we're going to go back and look in more detail at the chromosomal disorders, which we have already determined can be diagnosed using a karyotype, a picture of the chromosomes. These disorders involve problems with an entire chromosome, and the chromosomes may contain thousands of genes, so they're typically quite severe. Please remember that individuals who have one too many or one too few chromosomes are said to have an aneuploidy, an abnormal number of chromosomes. This results when there is a mistake in meiosis. Look carefully at the picture. In normal circumstances, sperm has 23 chromosomes, egg has 23 chromosomes, so the resulting zygote after fertilization would have the normal 46 chromosomes. When there is a mistake in meiosis, you might have a normal sperm with 23 chromosomes, but an abnormal egg with 24 chromosomes. This means after fertilization, the zygote has 47 chromosomes, which is an abnormal number or an aneuploidy. And every cell of that individual would have that abnormal number. Now we're going to go back and look at some of these chromosomal disorders specifically. So if you will locate in the upper right corner of your graphic organizer the space that says autosomal aneuploidy. The first autosomal aneuploidy that I want you to make note of in that space is Down's syndrome. In parentheses, beside Down's syndrome, please write trisomy 21. Now look over at your karyotype picture. On your karyotype picture, locate position 21. You will see on that position that there are actually 3 chromosomes rather than the normal 2 chromosomes. Circle that. You can see that it's also circled on the slide. Label that circle trisomy 21. This is an indication that this individual suffers from Down's syndrome. The reason this is an autosomal aneuploidy is because the disorder is a part of the autosomes. Remember, the autosomes are the first 22 pairs. Down syndrome leads to mental impairment, heart defects, flat facial features, and an enlarged tongue. These individuals can have a very severe form of the disorder or a milder form of the disorder. They often can attend school and get jobs. In many cases, you may have seen some of these individuals at our own school. Next, you are going to record some examples of sex chromosome aneuploidies. So once again, in the upper right corner of your graphic organizer, find the location labeled sex chromosome aneuploidy, and there are 2 that you will be writing in that place. The first one is Turner's syndrome. In parentheses, beside Turner's syndrome, write XO. The reason you are writing XO is because these individuals only have 1 sex chromosome. They have an X. So if you look at the karyotype on the slide, you will see at position 23 there is only 1 chromosome. That's why we record the genotype as XO. These individuals are 100% female. They don't have a Y chromosome. However, they are infertile. They are not able to have children of their own, and they often have some problems with their mental abilities, as well as some of their other body systems, as shown in the picture. The last chromosomal disorder you will record will also be a sex chromosome aneuploidy. It should be recorded as Klinefelter's syndrome. This appears to be covered up on the slide. K-L-I-N-E, Klinefelter's syndrome. In parentheses, please write XXY. Now look down at the karyotype on the slide and look at position 23. You will see that there are 3 sex chromosomes, an X, another X, and a Y. These individuals are 100% male. They have a Y chromosome. They do not have female parts. However, they are typically mentally impaired at least a bit. They are infertile, and they often have some characteristics such as mental fatigue, enlarged breasts, and a lack of facial hair. The last thing that we need to take a look at is way down at the bottom of your graphic organizer, the idea of gene therapy. Now, as I mentioned, sadly, most genetic disorders that we have talked about, all genetic disorders that we have talked about can be treated in many cases, but not cured. However, there is some hope for the future. This hope has arisen because of the Human Genome Project, which you have previously learned about. In the Human Genome Project, scientists have mapped the genes of a human so they know where genes for specific traits are located on specific chromosomes. And this has given us information that allows for the development of gene therapies. In the space provided, you need to write the definition for gene therapy, replacing a defective gene with a normal gene. So let's look carefully at the picture. Individuals who have cystic fibrosis are the ones who have an abnormal amount of mucus production. There is a gene that causes this, and the location of this gene has been identified by the Human Genome Project. The idea is to take a normal gene from a donor, put that normal gene into some sort of a vector. A vector is a carrier. In this case, they're using a virus. That virus is then delivered to the affected person via a nasal spray, for example. The idea is that the virus would then go and affect the cells in the lungs, delivering the normal gene to those cells. If this works, then those cells would begin to produce the normal protein rather than the defective protein that caused CF. This is still experimental. In fact, patients who have agreed to participate in clinical trials have even died from the treatment. But there is a lot of hope for the advancement of this type of treatment so that in the not too distant future, there may actually be a cure for some of these genetic disorders.